Yeah, today I'm going to tell you sort of more details on the Neuropixels 2.0 probes, and then I'll also tell you about our latest work with Neuropixels Ultra, which I think is informative in terms of understanding the kinds of things that we observe with Neuropixels probes and how we can push the limits of data quality going forward. So for the Neuropixels 2.0 part, I'll probably repeat some things that you might have heard yesterday here and there, but hopefully I can also add some things that weren't discussed. So Neuropixels 2.0 probes are designed for long-term stable recordings in mice. That was the sort of big picture design goal of Neuropixels 2.0. And in thinking about what would make an ideal probe for long-term stable recordings, long-term here meaning chronic timescale, the main feature that we wanted to achieve was reduction in the size of the probe. So literally the probe is decreasing about a factor of 3 in terms of the area of the base, the area of the sort of rigid part of the probe, and also in terms of the area of the head stage. That reduction of size by about a factor of 3 sort of magically, thanks to the engineering work at IMEC, came without cost in terms of the number of channels. So it's still 384 channels that can be recorded. So, we might have wished that the channels would go up, and maybe in future versions of the probe that will be the case. But here we got a size reduction without any reduction in channels. And the probe has 4 shanks rather than just one, or at least there's an option of the probe with 4 shanks. And in a few slides, you'll see why that particular feature, well, first of all, is I think good for many kinds of data collection scenarios, but especially for long-term stable recordings, giving you many, many more recording sites to access. So in addition to the size reduction and the number of shanks, it's not only the case that the head stage is smaller, but actually you can put 2 probes onto a single head stage and therefore get 768 channels on your single thin wire tether. And together, 2 probes plus a head stage weigh just about 1 gram. So that's quite good for small animals such as mice for chronic implantations in terms of the weight restrictions you might have. And the other design feature that changed is one that Marius alluded to. So with the Neuropixels 1.0, we had this sort of staggered site arrangement. The site sizes are identical in 2.0. They're 12 by 12 micron individual sites, just like in 1.0, but they're aligned vertically and they're also squeezed together just a little bit. So the distance from this pair of sites down to this pair of sites was 20 microns along the length of the probe. In 1.0, it's only 15 in 2.0. And so that means this sort of a vertical, sort of, if you want to count the edge-to-edge difference between probes that are vertically aligned, sites that are vertically aligned, that'd be 28 microns in 1.0, and it's only 3 microns in 2.0. And that accounts for the significantly better drift correction performance that Marius described. Yeah, I just wanted to put down here, before I move on, the list of authors on this project. This was a huge project, a huge collaboration. Matteo and Tim were the sort of leaders of this project, both in terms of the funding and the scientific direction and the wisdom and vision, et cetera. And also, the sort of those of us with our boots on the ground up here at the top were a team of co-first authors. Marius for spike sorting work, Anna and Bush for the chronic recordings and analyses, and Chatai also for chronic recordings. So yeah, this was really a big team effort, and yeah, it was a great experience to work with all these folks. Okay, so more details about 2.0, let's go. So here's what the probe looks like in the final iteration. So you'll actually note, if you look closely, when we published this paper, we had this sort of arrowhead design. That's changed just a little bit. There's a slight sort of streamlining improvement that IMEC was able to make for the final iteration. And this version that you see here, comparing 2.0 to 1.0, this is the final version that is on sale. Of course, I think you probably all already know, but I'm happy to say that you can go on IMEC and IMEC's website and buy this probe now. So we're very happy to see that happened. And the data quality is still high. So with the test version of the probe that we had with this arrowhead design, there was a very slight increase in the noise levels as the data acquisition switched from split band to broadband. But overall, the acquisition was still high signal to noise ratio. And I think it's the case that the final production version doesn't even have that increase in noise levels. So what I mean by split band versus broadband, so that's what we can see here. So in the Neuropixels 1.0, if you're used to recording with those, you might be aware that the high frequency action potential band, so sort of above 300 hertz, the band of activity, the band of signals that has spikes is split on the probe from the low frequency LFP data. And so you can see in this signal from Neuropixels 2.0 that in fact, those two bands of signals, the whole wideband signal is all combined into a single data stream. So you get just one data stream off the probe that has both your spikes in it and your local field potentials in it. And this is, I think, not too big of a impact on practical outcomes. I think you could do the same science with both versions of the probe, but certainly for some people in some processing pipelines, having the wideband altogether from the beginning might be a nice convenience. Here's some example waveforms that we recorded. I think these were recorded in my lab at the University of Washington, and just showing that we can record high quality, well isolated single neurons. Probably none of you will be surprised to see things like this, but just sort of confirming that the data quality is really nice on these probes. And here's the real payoff slide in terms of what was achieved with going to this smaller and multi-shank design. So this is data from Chatai Aiden in Seb Hessler's group. And the experiment here was that Chatai implanted chronically and permanently, but actually I don't want to say permanently, he implanted chronically two probes, one in the left hemisphere and one in the right hemisphere. And they go, each probe goes through cortex, hippocampus, and thalamus at approximately the same ML coordinate. And in this mouse, when the mouse woke up and was freely walking around, Chatai was able to record from over 6,000 recording sites. So how did we get to 6,000 recording sites? So first of all, not all 6,000 were recorded simultaneously. So again, each probe can still only record 384. So between the two probes, you can record 768 simultaneously. And so what Chatai did was he recorded, this group of 384 sites in the deep part of the left hemisphere and thalamus, simultaneously with this group of 384 sites in the deep part of the right hemisphere and thalamus. And then after making a short recording with those two banks of sites simultaneously, he switched, which is just a software operation to switch which channels he was recording from, to record, say, from this one, and then recorded for a few minutes from hippocampus and cortex on both sides, and so on and so forth. So there were 8 sequential recording sessions in this to make up this plot that you see here. So 768 sites at a time, 8 sequential recordings. And this recording then was from about 6,000, a little over 6,000 recording sites in this freely moving animal, not all simultaneous. There are actually about 10,000 recording sites altogether. So with the 1.0 probe, you might remember there's about 1,000 recording sites on the probe from which you can select which 384 you want to record. On 2.0, because there's 4 shanks, you're getting a multiplier of 4, but you're also getting a bit extra multiplier because of what I mentioned before, which is that the sites are a little bit closer together. And so that gives you about 1,200 sites per shank. And so then 1,200 times 4, we get up to about 5,000 sites per probe, about 10,000 overall. Okay, hopefully all those numbers are making sense and you can see how this data was acquired. And I think then this sort of brings home this point about how the multi-shank is particularly useful maybe particularly useful in this sort of chronic cases, because if you can go back and record day after day or block after block from different sections of the probe, then you can really actually assemble a data set in which you're sampling thousands and thousands of recording sites. Again, not all simultaneously, but you would be able to sample the neural activity at all of those points in the brain. You can also use different kinds of channel configurations. So here's a different example in data from Josh Steinmetz's group, collected by Gentle Park. They instead of selecting a bank of 384 sites on a single shank, in this example recording, Gentle selected 96 sites per shank, So that gives about 3/4 of a millimeter span of recording on each of the 4 shanks all simultaneously. So still 3D4 sites, 96 per shank. And that gives you this sort of little 2D postage stamp recording, which is a really nice geometry for some brain regions, like the dorsal striatum in the mouse, where that sort of fits nicely in the span of dorsal striatum, and they could record a large number of neurons in dorsal striatum. Here they were sort of delighted to see that they could see on individual trials these sequences of spiking activity, which it's a bit hard to get an intuition for what this would look like if you had only a third of the number of neurons. But that is what they had previously been getting with Neuropixels 1.0, is more like 50 neurons instead of 150. And in that situation, they weren't really able to sort of visualize these sequences of spiking as clearly as you can with the density achievable, the local density achievable with Neuropixels 2.0. So what kinds of site selection? So I've mentioned 2 different kinds of site selection patterns. So I'm just going to have one slide giving a sort of brief overview. I'm not going to go into too much depth here. So this is the figure from the manual, at least the initial version of the manual that we had. And what you can see here is these groups of sites along the shank have these colorful little numbers, which correspond to which group of channels the sites connect to. So here I'm using the word site to refer to the physical location on the probe that is collecting data, and channel to refer to one of the 384 recording 384 lines in the recording apparatus that are accessible to collect data from a site. So a given channel can connect to multiple different sites. And so, for instance, the channels that connect to these sites down here on this deep part of the left shank, those same channels can also connect to these sites or these sites or the logic here. And so then you could, in principle, sort of, and then the point is that you're not going to be able to record from the sites that are mapped to the same channel. So you simultaneously, so you cannot record from, say, these sites and these sites simultaneously. That won't work because that would be a conflict on the channels. Hopefully that logic makes sense. You're probably not going to ever sort of sit down with this complex little diagram here and try to work out some fancy pattern, because I think there are just a few patterns that are useful for probably just about every case. So one of them is what I showed you in the data from Josh Steinmetz's lab, where you select just the deepest 2 groupings, so the deepest 96 channels on each shank. So that gives you this nice little 2D patch that I mentioned on the last slide. Another is where you put all of your selections onto a single shank, and you can do that on any of the 4 shanks, as you can sort of verify by looking at these. And so that gives you sort of a single shank recording, and that was what Chetai used in the chronic recording a couple slides ago. There's a couple of other example configurations here, including a sort of diagonal one. And then the one other configuration that we like to use for some things in my lab is one that looks like this. So I made it even more confusing here because I've split each of these color groupings in half. And what this is indicating is that I select the odd numbered channels from group 1 to be mapped down to this part of the probe, but the even numbered channels from group 1 to be mapped up to this part of the probe. And so what you're achieving here, it's still 96 sites per shank, but if they're at double the span and half the density relative to this configuration. So here you can cover 1.4 millimeters, but only with a single sort of vertical column of sites. So that's another configuration that can increase your sampling spread a bit at some cost to your ability to resolve individual nodes. Hopefully that makes some sense. Okay, so here in data from Mush and Anna, they performed chronic implants. And so in this example plot and these example waveforms, Mush and Anna were really testing, what is the limit here? Like how long can we have good high success rate chronic implants? And you can see here we went out almost to a year. So this is day 309. So almost a year of recording where the sort of band of spiking that you see, the bands of spiking that you see in cortex and hippocampus and in thalamus are really quite well preserved over that time. This is just one of these. This is not exactly the same as the drift maps that Marius was showing you, but similar logic where y-axis is the depth on the probe. So you can see there's definitely some sort of slow instabilities in both in terms of the firing rates and also a little bit in terms of the depths. But generally, we're recording from the same regions and bands of spiking activity across all of this time. So this was really exciting. I mean, I think one thing I just want to say here is I get asked a lot about, neuropixels versus flexible electrodes. Of course, there have been a number of really nice papers about using flexible electrodes, and that technology has been adopted by groups like Neuralink, Elon Musk company, doing brain machine interfaces. And I think the reality is that at least for rodent experiments, we have really had pretty great success just implanting and recording over long time scales with good quality data retention. So the argument for the flexible electrodes is that okay, they'll come at a cost of being like lower channel count and also really difficult to do the surgeries and really difficult to insert, but you're supposed to get higher quality data for longer periods of time. And I don't, have any like physics explanation for you about why we can see these results that we do, but our literal observation is just that we can record for up to a year. We also showed something quite similar actually in the Neuropixels 1.0 paper. So this is an observation that has been replicated with good data quality for that time scale. And here's sort of a similar kind of quantification, but a bit more summarized over many different experiments in both rats and mice in 6 different labs, showing the total firing rate that's recorded over a 2-month time period. So this is a zoom in on some of the axis here. So a 2-month time period, and also the neuron count that was obtained over those time periods. And you can see that definitely there's a slight decreasing trend on some of these recordings, more so maybe for the neuron count than for the total firing rate. So there's some variability from recording to recording. This was probably one of the good examples that would look quite stable. It should be one of these examples that looks quite stable in these plots, and other ones can have more of a declining. So there is variability, but you can achieve really good results. And overall, I think these are quite satisfactory results for doing most kinds of scientific questions that people like to ask with rodents. Okay, so I'm just going to tell you briefly about how in this paper we approach the question of asking whether the same neurons can be recorded across days. And I do understand that you'll have much more updated state-of-the-art description of these issues later today. So this is really just describing what we had done at the time of the Neuropixels 2.0 paper with the data sets we had then. But I think it's worth sharing what our observations were. So again, Mush and Anna recorded these chronically implanted probes across days, at least 2 months on multiple different probes. And they observed that you could see the same sort of patterns of spiking shifting from one day, here day 26, to another day, day 36, as if the brain had just moved a bit from one day to another. And so that suggests that we could use the kind of methods that Maria showed you to do some drift correction across days and successfully resolve the identity of, say, these bands of spikes as being the same neuron. And to validate this, Mush and Anna did a really nice manipulation, which was to show on each day of reporting, they showed a series of natural images to the mouse, which were the same series of natural images. So this little rat in a palm, mushroom, etc., were all shown on each of different days. So for example, for this neuron, day 14, 15, and 24. And so then we could use that as a kind of a ground truth. We could say, okay, if it's the case that the visual response signature, what we call fingerprint, looks the same for a given neuron that's returned by the spike sorting algorithm after attempting to stitch together the probes, if that visual fingerprint looks consistent, as it does for neuron 19 here, it only responds to the mushrooms. And as it does for neuron 9 here, it responds mostly to image 90 and less for the mushrooms. Then we can conclude that the algorithm must have done a good job at sorting the correct neurons together. And so this is a kind of summary of how that looks. So here we're just taking 2 adjacent days, day 14 and day 15, and we're asking, for each neuron that was returned by the algorithm, which existed on both days, day 14 and day 15, did its signature, that visual fingerprint, best match the spikes that were sorted together on the 2 days, or did it best match a different set of spikes. And the fact that this looks like a straight red line is because for almost all cases in this example where they're separated by just 1 day, the visual fingerprint was matching between the 2 units that were sort of together. There's just one example right down here, if you can barely see it, where that was not true. And these 1, 2, 3, 4 gray boxes are the 4 shanks of the probe. If we look at a greater separation, day 15 versus day 36, first of all, there are fewer overall units, which is because some just drop out in their spiking. And also the matching is a little bit less successful. Certainly you have more examples where the visual fingerprint doesn't match correctly here. So this just summarizes that kind of analysis for many different mice in many different sessions. And so what we can see is the time between recordings on the x-axis, the estimated percentage of stable units on the y-axis. And you can see there are quite a lot of these right at about 100%. And then there are others that have less success as estimated by this visual fingerprint analysis. And so on average, what this looks like and the way we kind of summarize it intuitively for ourselves, the conclusion that we made was that you can record about 90% stably over this 2-week period and about 80 stably over the 2-month time period. So this, has some sort of average curve like that. And certainly this is these are numbers that are a function of both the data quality and a function of the processing steps. And so it may well be the case that, hopefully we'll hear that this is an improved process now with more updated algorithms. Okay, and so I think we're also going to hear about chronic implant designs, or maybe you heard about yesterday. But we did. I just do want to show the one that we included in the Neuropixels 2.0 paper from Sid Hessler's group and Chetai Aydin, which is a really nice chronic implant design that makes the probes recoverable. And so this is just to show briefly this example, one of these chronic implant designs, but also just to say that Chetai was able to recover probes and then reimplant them and have similar spike rates and similar numbers of neurons recovered from the reimplanted probes. So it appears to be the case to us that when you recover a probe, assuming you clean it properly and assuming nothing goes wrong with the explantation and reimplantation processes, then you can actually have data that are just as good as the initial clean probe when you do a reimplantation of a probe. So again, this is a characteristic that makes these probes really nice for these chronic long-term recordings. All right, the last thing I want to say about 2.0, I think, is that there is another version of it that's going to be coming out probably next year. I understand that they're close to having this ready. And certainly my lab is very excited. We don't have any of these yet in my lab even. So we'll be as excited as all of you will be about this version. So if you're doing acute recordings, maybe you don't care quite as much about the svelte narrow size of the 2.0, and maybe you're willing to have a broader base. And if so, there's this probe version called Neuropixels 2.0 Quad, which is a 4-shank probe that is essentially 4 Neuropixels 2.0 probes stacked together, essentially left to right. And the advantage here then is that each one of the shanks has 384 channels to record from. And so in total, then a single probe has 1,500 recording channels of data that it can acquire, which is really exciting. So it would mean that, to flip back briefly, you don't have to make these kind of trade-offs between the same kind of trade-offs between span and density. You could just have all 4 shanks have the 384 channels. So that's really exciting. But again, at a cost to the device size. Okay. I wonder if I should do, well, okay, I'll just carry on. Yeah. So gears and talk about Neuropixels Ultra. So let me talk about some of the remaining challenges that we were thinking we might want to try to address with a sort of a new direction research project. So first of all, the yield of spike sorted neurons. I say it is low here, maybe that's not generous enough, but we have this concern or consideration that we get, you insert a Neuropixels probe, you get 100, 200, maybe 300 neurons, and is that all the neurons that were near the probe? Are we missing something? You can do a back of the envelope calculation and convince yourself that there probably ought to be more neurons near the probe than that number. And so one might wonder whether there are some neurons that we're not observing well, or whether we're not capturing the space of neurons near the probe effectively with Neuropixels 1.0 and 2.0. So the yield on spike supporting neurons is lower than we might expect it to be. Small neurons and processes are difficult to record. So, this is a cerebellar granule cell, famously small and difficult to record extracellularly cell type. And even with 1.0 and 2.0 probes, people have had, let's say, limited success in recording cell types like cerebellar granule cells. And so we imagine that there might be some types of small neurons, or even processes of the neurons, like dendrites, that might be difficult to record with current versions of the probes, like 1.0 and 2.0. And finally, you have minimal cell type information when doing extracellular electrophysiology. So specifically, what I'm showing here is broad and narrow spike waveforms. In red are these narrow spike waveforms that have a sort of a faster time course to repolarization, and in blue are some other neurons that have broad spike waveforms. And this characteristic of the waveform, essentially, the way you can think about this is that it gives you one bit of information about the biophysical characteristics, and therefore the cell type of the neurons that you're recording. You get one piece of information about whether they express some particular channel that causes them to repolarize quickly or not. And that is some cell type information. So it's great. And I think it's been used many, many times in the literature to great profit. But I think that we would really like it to be the case that we have even more cell type information in extracellular electrophysiology. So these are the kind of challenges we were thinking about, the kind of limitations of extracellular ePhys that we were thinking about when we designed the Neuropixels Ultra probe. And we had the idea that there might be a single kind of change to the probe design that could help solve all of these challenges. And so that's the ultra high and dense, ultra high density and small site size that you see here on the right for Neuropixels Ultra. So again, this is just comparing the site patterns for 1.0, 2.0, and Neuropixels Ultra. Again, with 1.0 and 2.0, they have these 12 by 12 micron sites, but aligned and a little bit closer together for 2.0, giving a total site density of 100 sites per millimeter and 133 sites per millimeter for 1.0 and 2.0. Whereas for Neuropixels Ultra, the sites are much smaller. They're 5 by 5 microns with only 1 micron gaps in between sites. So that gives this 6 micron site pitch, or center to center spacing. And that increases the density by a factor of 10. So from 133 to 1,333. And you can see a sort of electron micrograph of these probes zoomed in and see these tiny recording sites. So before I even go any further, let me just point out the other part of the graphic that you're seeing here, which is that, of course, the big trade-off here is that there's a reduction in span. So if you increase the density of sites with the same number of recording channels, the 384 recording channels, you're going to have much smaller span. So whereas you get about 3 millimeters for Neuropixels 2.0 and almost 4 for Neuropixels 1.0, there's only a fraction of a millimeter, about 300 microns, that you can record with Neuropixels Ultra. And again, like with the Neuropixels 2.0 project, I want to point out that this was the work of a really big collaborative team from multiple different labs. And I, in particular, want to highlight the work of Joanne, who's a postdoc in my lab, and Andrew Shelton, who's a research scientist at the Allen Institute, who sort of spearheaded the data collection efforts and data analysis efforts that I'm going to tell you about on the coming slides here. Okay, so to give you another sense of the scale of Neuropixels Ultra, here's just a cool to-scale overlay of an individual mouse visual cortex pyramidal neuron overlaid on a Neuropixels Ultra recording grid, just so you can see the comparison of exactly what kind of spatial scale we're talking about when we talk about this ultra high density. It really is the spatial scale of dendritic processes and features of the morphology of neurons. Okay, and here are a couple of example waveforms showing the kind of thing we can observe, the kind of thing we just can see with these probes. And so, we kind of think of these probes as almost a different class of device than Neuropixels 1.0 and 2.0. Maybe this is overstating the case, but you can really think of this as like an implantable voltage camera, right? You're like imaging the voltage fields over time, and that's what we're playing out as a sort of a movie here of the mean waveforms of a couple example neurons, one in thalamus and one in striatum. And hopefully you can see that there's some sort of definitely non-trivial features to these waveforms. You can see different spatial zones of the potentials and even some sort of different propagation directions. Maybe you can see that there's apparent propagation often this direction as well as often this direction from the striatum neuron here. So there's definitely, we think just by looking at these example waveforms, some more complexity observable. And I'm going to show you just a couple other example waveforms in a slightly different plotting format, just because I'm going to come back to this plotting format a few times throughout the subsequent slides here. So here's one example neuron and another example neuron. In this plotting format, we're showing on the one hand just a simple grid of the grid of recording sites as a spatial map. So the axis of the probe and the axis across the face of the probe. And this is just showing the amplitude of this across these different sites. And then on the lower plot is the time course of the waveform of the unit on the peak site and on some adjacent sites in gray. And then in this blue and red one, we have the spatiotemporal aspect. So taking the peak column of sites and plotting the temporal waveform on each of those sites along that column as a color map. So you can see that allows you to see that, whereas this example waveform is kind of stationary, this one has a sort of a propagating feature going up the probe. Okay. So Neuropixels Ultra sacrifices span for density, as I mentioned, and the smaller recording sites come at a modest increase in noise levels. So this is a plot showing the expected change in noise levels from Neuropixels 1.0 with its 12 by 12 micron sites. So that's a electrode site area of 144 square microns down to Neuropixels Ultra with 5 by 5, so 25 square microns. And you can see that the total noise should definitely increase, but it's only an increase of a few microvolts RMS. And so we think of this as pretty minimal or modest impact on data quality. And you can see that summarized for data recorded in vivo and in saline here. So we did introduce a new spike sorting algorithm in this paper. I'm only going to describe it briefly. It comes from Liam Paninsky's group. Julien Boussard led the application of this application and tailoring of this algorithm to the Neuropixels Ultra datasets. This was also developed with Charlie Windolf and Cole Hurwitz in Liam's group. So I'm just going to describe a couple of the features of what DartSort is doing. I guess I want to emphasize, especially having followed Marius here, DartSort is the code is available. I think you can download it and probably succeed in running it. But it really hasn't been tested on a wide range of data sets yet. So I don't think at this stage I would expect that it would outperform Kilosort for Neuropixels 1.0 and 2.0 data sets. I would definitely be planning to stick with Kilosort for those, at least until DartSort has more thorough kind of testing of the kind Marius showed. So it does have some cool features, though, that are particularly tailored for these kind of high density probes. So the first thing that DartSort does is detect and localize individual spikes and do motion estimation. This step is quite similar to what Marius showed, where you're sort of like figuring out the motion over time. And you can see we did the same kind of manipulation with the imposed zigzag motion in these data sets. There are some differences to exactly what is happening here. I'm going to skip over them. The main difference in what DartSort does is that DartSort then attempts to do that initial clustering. So Marius described a process where you have an initial clustering with simple features followed by template deconvolution based on the templates determined from that clustering. And this initial clustering with DartSort is done in spatial domain. So because we have this dense grid of sites, we can actually spatially localize each individual spike after denoising the spikes to be able to observe that there are these spatial clusters, sort of similar to what Marius showed you from the Neuropixels 2.0. data sets. So between space in depth along the probe and across the face of the probe, as well as just the amplitude of the spikes, you can do, excuse me, an initial clustering that separates out the spikes that putatively come from the same neurons. And you can determine from these the templates, and in particular, this uses something that Marius also alluded to, which is a sort of super resolution template discovery, where you're taking the spikes that occurred naturally at different depths and using those to determine what the waveform looks like at all different depths. And then you can go back through the data when you're doing your template deconvolution to find the most accurate and most precise version of each template waveform. So these are the kind of steps that DartSort implements. Again, I'm not going into much detail here. And again, I don't really recommend at this stage that you go out and try DartSort on Neuropixels 1.0 and 2.0 datasets. But I would keep my eyes out for it if I were you, because there's a paper under review, peer review about DartSort right now. And I know that they're working really hard to sort of perform some of the kinds of comparison analyses that Marius was showing you between DartSort and other sorting algorithms. So we'll be quite interested to see how it does. Okay, so we use DartSort, and I'm going to quantify then a few different metrics about how the neurons look and is Neuropixels Ultra Pro. But to sort of understand what Neuropixels Ultra is buying us relative to other geometries, relative to 1.0 and 2.0, we used this strategy. We used a strategy in which we took the original Neuropixels Ultra data and spatially resampled, so sort of a kind of downsampling, to make a data set that looks like it was recorded from the exact same neurons, but with a 1.0 or 2.0 probe, or with what we call a large dense probe. So let me just describe what's happening here. So we take the Neuropixels Ultra data set, which is 8 sites across by 48 sites long, and we take groupings of those sites and average them together so that these 4 sites on the Neuropixels Ultra become one site in this new constructed resampled data set, which is Neuropixels 1.0 like, in that it has the kind of zigzag pattern at broad spacing. The spacing is not exactly equivalent to Neuropixels 1.0 because we wanted to keep on our Neuropixels Ultra grid, but it's quite similar. Same thing, you get the idea with Neuropixels 2.0, they're a little bit denser and aligned. And then this large dense is just averaging every 4th site. So we're including all the data. There's no gaps in the coverage here, but we do have the larger recording sites rather than the tiny recording sites of Neuropixels Ultra. And so here's just an example of what that looks like in practice. This neuron had a really small waveform. It existed on just a few channels in the original Neuropixels Ultra data set. And this is the waveform on the peak channel and a few adjacent channels. And when we do this spatial resampling, in this particular case, we happen to miss the peak channels of the particular neuron. And so when we do the Neuropixels 1.0, we get out a new mean waveform that is what we would have seen from this neuron if a Neuropixels 1.0-ish probe was located at this location. And that new waveform has a much smaller peak amplitude in this case. And so this is using that strategy to quantify a few features: the amplitude, the signal to noise ratio from Neuropixels Ultra and from these different configurations, the large dense, the 2.0 and the 1.0. And you can see that the amplitudes we record on Neuropixels Ultra are higher. And that's more than compensates for the increase in noise level that I mentioned earlier. So when we compute the signal to noise ratio, we also get a significant increase in Neuropixels Ultra relative to large dense 2.0 and 1.0. So given that each individual neuron has a higher signal to noise ratio, you might imagine that if we take the spatial resampling strategy and apply it to the raw data and rerun spike sorting on these new raw data sets, that we might get a higher yield from Neuropixels Ultra than from these comparison lower density data sets. And indeed we do. So here, the height of the bar indicates the yield for 3 different definitions, sort of 3 different stringencies of what we determine to be yield. So in the tallest bar, you're seeing just all the count of units that had a stable firing rate across the recording session. In the next highest bar, in the next darkest color, you're seeing the units that had a stable firing rate, but also a stable visual response. So we did the same kind of fingerprinting here, and we checked the fingerprint of the units at the beginning of the recording session to the end of the recording session. And so, these average 33 units from Neuropixels Ultra that fall in this darker bar, they had both a stable firing rate across the session, but also their visual fingerprint was very similar from the beginning of the end to the end of the session. And then here we have a stringent metric on the refractory period violations to make what we would call these sort of like high quality isolated single units. And you can see that is certainly reducing the yield a lot on all probe designs, but still more for Neuropixels Ultra versus 1.0 and 2.0. And the difference here between Neuropixels Ultra and large dense is not significant. So overall in the yield, the large dense is indeed quite similar to Neuropixels Ultra, but there's a big hit for 2.0 and 1.0. And you can see that quantified sort of as differences with error bars across the data sets that we sampled. And here, these numbers are just summarizing. If you look at the stable units with stable firing rate and visual responses, you have about a 3x increase of Neuropixels Ultra versus 1.0. That's this bar versus this bar, and about a 60% increase even if you look just at the high quality single units. That's this bar versus this bar. So this is a big increase in yield. It's a relatively more modest increase in yield if you're thinking only about the highest quality, well-isolated single units. The 60%, of course, anybody would take 60%. Maybe you wouldn't take that at the cost of the span that you're sacrificing in this particular probe. So that would be a question for your particular application. One more thing I was going to say about this. Oh yeah, so sort of the inference here we have, because the number of well isolated high quality single units is relatively less increased, our inference is that most of what we're gaining with the Neuropixels Ultra are the relatively lower amplitude units that are being sort of failures of detection or failures of having just quite enough signal to noise ratio under the Neuropixels 1.0 and 2.0. So you're getting more and more low amplitude units that you can successfully spike sort out, as opposed to failing to detect them or failing to successfully spike sort them with the 1.0 and 2.0. So a high amplitude large unit, you probably record it just about as well on both probe types. Okay, so now I'm going to tell you a bit about some of the kind of biology we observed with these probes, and that I think help us understand both, what we see when we make electrophysiological recordings, but also what kinds of things you can do with technology like this. So some extracellular action potentials that we observed have really small footprints. So what we're going to call a footprint. The footprint is just the measure of the radius over which you can see that neuron with significant amplitude. That's how we've defined it. And so you can see an example such unit here. And the remarkable thing about this neuron is that it has quite a large spike amplitude at its peak site, but then within 12 microns, so just a couple sites over, this neuron has disappeared almost entirely. So this is pretty striking because not all neurons have this property. Most neurons you see over quite a wide spatial range. And this is the kind of neuron that's small enough to really, this kind of unit, I guess I should say, that's small enough to really fall in the gaps between sites of a Neuropixels 1.0 or 2.0 probe. So we wanted to try to understand where we see these and what they might reflect. So Joanne made these recordings from lots of different brain regions. You can see the recording tracks in orange here. And here are some example neurons that Joanne recorded from visual cortex. This is that plotting format I mentioned, where here you see a large spatial span neuron. So this neuron has a footprint of 90 microns. This neuron would be easy to pick up on lots of sites from 1.0 or 2.0. Whereas this neuron is an example of the small footprint neuron I showed you. It has this little tiny footprint just on a few sites, even though the peak amplitude is quite comparable between the two units. And here is the distribution in visual cortex of the mouse of the footprint, where we can see there's almost a second mode, and you'll see this more clearly later, that this really is a second mode of this distribution that have of units that have really small footprints. And so we looked across many different brain regions, and we had quite similar observations. We see these small footprint units in CA1 hippocampus, in dentate gyrus, in CP, this is dorsal striatum, in VPM is a thalamic nucleus, in corpus callosum, we see quite a few of them. And to summarize these observations across regions, this is the percentage of small units in all these different regions. And sort of surprisingly or strikingly, it's a pretty similar percentage. It's about 10% in most areas we reported from, with the exception of mainly two, dentate gyrus and corpus callosum. And so that's interesting because, of course, corpus callosum is axon tracts. So there, where we record small footprint units in corpus callosum, one could quite probably confidently and reasonably infer that those correspond to axons there. Dentate gyrus is interesting because it's a brain region that has small and dense cell types, so these dentate gyrus granule cells. And so one could wonder whether this corresponds to recordings from those dentate gyrus granule cells. Before I speculate about that more, let me also show a comparison across different species. So we were excited to have data from macaques, from electric fish, and from bearded dragons. And we again saw the same kind of thing, a broad distribution of spatial footprints recorded, and in particular, a fraction, a sort of reasonably similar fraction of units that had small footprints in all of these species. So we really see these small footprint units everywhere. So what do they correspond to? So as I mentioned, presence in corpus callosum suggests that axons is one possibility. Presence in dentate gyrus suggests that small cell type is another possibility. And so we made a hypothesis that we sought to falsify. So we hypothesized that in neocortex, starting in one place, they may correspond to a particular interneuron type. So this is taking this small cell type hypothesis and sort of writing it down formally so we can attempt to falsify it. So how do we want to try to falsify it? Well, first of all, we noticed a particular property in neocortical units about their footprint, which is that if you plot the duration of the units, so the spike width against the footprint, you see this quite interesting distribution where the majority of units have large footprint and large duration. But among those with small duration, that is the narrow spiking units, there is a really clear double mode where you have some that have large footprint and some that have small footprint. So in any case, most of these small footprint ones are fast spiking. And so we, maybe we took this as kind of additional probability of support for this small cell type hypothesis. We thought, okay, maybe it is the case that these small footprint units, fast waveforms, perhaps they correspond to PV positive inhibitory interneurons in neocortex. So to test this, we did optotagging. So we expressed channel rhodopsin in PV positive interneurons. We made recordings with Neuropixels Ultra, and we elicited spikes from a subset of neurons recorded on the probe and inferred those to be the PV positive ones. And so again, here's this distribution of all units and the distribution of just the footprints of just the fast spiking or just the regular spiking neurons in cortex. And here's the distribution for parvalbumin positive optotagged neurons. So here's two example neurons, and here's this footprint distribution on the same axis as this. And so you can see that PV neurons in general don't have small footprints. There's a small percentage of them that do, but in general, the distribution of PV positive inhibitory interneurons has large footprints, just like the distribution of regular spiking and fast spiking. So it doesn't appear that the small footprints with their narrow waveforms, it doesn't appear that they correspond to PV positive interneurons. And we checked two other lines, somatostatin and VIP. It was a little bit news to me, but there's already reports in the literature that some somatostatin positive and VIP positive also have narrow waveforms. So that's in general a caveat about using narrow waveforms. But in any case, we found that SST and VIP also have, in general, broad spatial footprints rather than small footprints. So none of these three inhibitory interneurons types can account for the small footprint fast spiking units that we see. And these certainly aren't all inhibitory interneuron types, or certainly not all cell types in visual cortex of the mouse, but these three classes do account for something like 90% or 85% of inhibitory interneurons in V1. So we think it's pretty unlikely then that these small footprint fast spiking units correspond to a particular inhibitory interneuron type in V1. We have another observation that kind of confirms that, which is we looked at cross correlograms between these small footprint units and other units that were recorded simultaneously. And here's ACGs from 2 example small footprint units, and cross-correlograms between the small footprint unit and another unit. And you can see that in these 2 examples, and we have more examples like this, that you can see either an excitatory type phenotype or an inhibitory type phenotype, where this looks like a monosynaptic excitatory or a monosynaptic inhibitory connection between the small footprint units and their postsynaptic targets. We conclude from this that small footprint units likely correspond to axons. We can kind of imagine that this may generalize across many of the brain regions we recorded, because part of the evidence is that they have a high prevalence in corpus callosum, and then the other part of the evidence is that certainly in visual cortex of the mouse, they don't appear to correspond to any particular neuron type. They can be excitatory or inhibitory. Okay, I think last slide. So I think I'm on time. So finally, we wanted to use that data set that we collected with the optotagged waveforms to ask whether Neuropixels Ultra could improve the cell type specificity of the recordings. And so in particular, we extracted all of these features, like the amplitude of the neurons, their spike width, the peak to trough ratio, repolarization slope, which is all of these things that you can sort of characterize the waveform of a unit by, and use them to decode the identity of a neuron as PV, SST, or VIP. And here's this decoding confusion matrix where you have the true cell type of the neurons on the y-axis and the predicted cell type on the x-axis. And you can see that the decoding is actually quite good. So it's about 75% success amongst these 3 classes, which is a chance level, of course, of 33%. So that, I think 75% is probably, you'd wish it were 100%, but it's probably good enough to do some kinds of scientific questions with. Of course, unfortunately, this isn't quite the scenario you find yourself in if you just stick your probe in mouse visual cortex and do a recording, because you don't know whether the neuron in the first place came from one of these 3 classes. And so here is attempting to decode amongst all the types that we recorded, both the optotagged units and the non-optotagged units. And so the total success here is lower. It's about 60%. Of course, in particular, like one of the kinds of errors that's quite common here is the large footprint fast spiking units with the PV units, because presumably most, if not all, of these large footprint fast spiking units are PV units. So there's some kinds of errors here that make this an underestimate. But in any case, there's about 60% correct, and we found that was better on the Neuropixels Ultra relative to about 40-45% on the Neuropixels 1.0 spatially resampled site probes. And we do a little better by including spatial footprint specifically relative to features that don't involve the spatial footprint. So part of this improved cell type specificity comes from having spatially extended information. Part of it comes from just having more likely having more samples from near the peak site of the individual units. Okay, so let me conclude. So Neuropixels probes enable large-scale electrophysiology, and what I've sort of argued here is that that's either a large span, as with 1.0 and 2.0, but can also be large density, either way, taking advantage of the large channel count. Using these smaller recording sites allows detection of smaller electrical fields, and we think these probably correspond to axonal recordings. So this, I think, is both it's most straightforwardly, it's an opportunity that if you are interested in recording from axons from a region, because you're interested in maybe more directly studying the inputs to that region, for instance, then this can be a tool that can do that. And there have been some really nice papers, one from Mateo's lab, from Sylvia Schroeder, and some from Jens Kremkow, profitably using Neuropixels 1.0 already to record axons in certain cases. And I think this technology improves the ability to do that in many brain regions. And finally, cell type specific information is available in extracellular EFIS beyond just broader narrow spiking. And I think there's a lot of improvement that will be done in this going forward. So again, I want to thank the really big team that worked on Neuropixels Ultra, as well as, of course, the 2.0 that I presented in the first part of this talk. Thanks to my lab, who contributed to this and other work. And thanks to funding and support from NIH, HMI, Wellcome Trust, and Allen Institute. And I'll take questions.